Well, welcome back to another public lecture. This one comes from Lincoln University, and it's a special lecture because this is their 2019 Christmas lecture. And the first lecture in this Christmas series comes from Dr. Fabian Payerson, who's a theoretical physicist, and he's presenting the many dresses of DNA.
Ladies and gentlemen, we're moving now to part 2. Before I start, let me just remind you, if you have your breakable glass drinks or any other glass drinks, would you please, after the talk, just leave it by the counter outside. And also, I'll still remind you that you enjoyed music today by Jack Brett and James Manners from College of Arts of University of Lincoln. And we move into physics. Don't forget to write everything because we'll be exam at the end. It was a joke. Our speaker, and again it's already tradition, those who who came here year before and year before, you know that our Christmas lecture delivered by two speakers, Professor Anna Marie Roos and Dr. Fabien Payerson. Fabien Payerson is a senior lecturer in the School of Mathematics and Physics, University of Lincoln. He holds a Master of Science and Doctor of PhD in Theoretical Physics from University Pierre-Marie Curie in Paris, in France. He came to Lincoln in 2015 from Durham University. He also worked previously at University of Cambridge and University of Barcelona in Spain. He publishes in varieties of fields applying physics into life sciences, material sciences, and DNA, of course. His broader interest in theoretical physics and computational modeling, foundational physics, physics in education, artificial intelligence, logic, as well as philosophy of science. And please give a warm welcome to Fabien Payerson.
Thank you very much, everyone, for being here. Thank you very much, Andre, for the introduction. I just remind you, there was this Padlet link before, so if any of you had recorded the link on your device, then please do ask questions as the talk is going. Now, I will be talking about DNA's 1000 dresses. Of course, the title is a little bit elusive, isn't it? And it turns out that my initial title idea was a bit longer. It was, how does the idea of different clauses for DNA help physicists make sense of DNA's biological function? You see, it's quite a mouthful. Before going into the physics, which is in the title of DNA, what we are going to do is take a step back and basically contextualize a little bit why I want to talk about DNA. So let's imagine two friends discussing how important DNA is in our current society. One of them would say, well, how could we check how popular DNA is in British culture? And of course, the mate would say, well, maybe by checking the usage frequency of the word. And of course, you need to thank this for this is a great idea. So how would we do this? Well, the way I did it was by looking at something called the British National Corpus. And it contains 100 million words out of 4,000 different documents, audio and written documents. And then you can look at how frequently the word DNA appears in there. Now, if you look at DNA, you will find that it appears 3,310 times. Now, of course, it's difficult to know whether this is a big number or not. So we need to compare it with something we are more familiar with. So I will compare it with a word which appears a little bit more, which is 3,316 times, but the word itself is beer. And so because we are in the UK, well, we can conclude that DNA is actually very popular indeed. So where does this particular popularity come from? Now, in 2003, Steve Connor highlighted 23 different ways in which DNA changed the world. And here what I would propose is basically 4 main arguments that emerge from this particular article. So the first argument is that it provides a molecular explanation for trait inheritance. Okay? The second one is that it unlocks new development in medicine, and of course we do want to live longer and healthier. And another one, which was related to what Anna Marie was talking about earlier, which is that it may hold the secrets to the origin of life on Earth. I'm being careful here, I'm saying Earth. And finally, in some sense, it also may enable a fair justice system. So that was in 2003 that Steve Connor made these arguments. Since then, few things have happened. In particular, these 2 things, which can be lumped together into the idea of new technologies that are beyond biology, but which make use of DNA. One of them being the one on the right here, which is basically storing information, like digital information with DNA. So that's why it's actually useful. Now, what is DNA? So Anna Marie talked about it a little bit. So this is deoxyribonucleic acid. For a physicist, it means it's complicated biochemistry. And I won't pretend that I'm able to explain all the intricacies of what it is, in that respect at least, in the chemical level. However, if I may present a simple, if not simplistic picture of DNA, then it will look like this, a kind of ladder with 2 different strips, like blue and red here. And the way the vocabulary would go is that this blue one here would be called a DNA strand. And in principle, it can exist isolated, like in a test tube, for example. And some people use that for technological purposes. And then this set of letters, Anna Marie talked about earlier, this is the genetic alphabet in which the genetic code is written. This red one here is called complementary DNA strand. And as you see here, again, related to, just taking from what Anna Marie said, just in fact revision, if you will, then you've got, it's a negative version of the initial strand that you've got in blue. What happens after that is that if you look at 2 consecutive pairs of letters, assuming they want to be as close to each other as possible, you could visualize them as being like a pair of dumbbells that you want to get as close as possible to each other. Now, of course, if you put them parallel to each other, you can only go so close, right? Because otherwise they will overlap, and overlaps are not good. So parallel pairs cannot get closer than this without overlapping. What you can do, of course, is to put a twist to it, and which is rotating slightly one out of phase, if you will, with respect to the other, and then you can then get them closer to each other. And then in that case, you get the very particular initial drive for the whole ladder to form an helix. So from this, you get this typical type of double helix, and that's the stable form in which we are used to talk about DNA and visualize it. Now, what can physics, for some reasons I missed up here, what can physics bring to the discussion? Because so far I'm talking about DNA. Now, I will try here to take a very broad perspective about it. So if physics is to be considered like any other creative activity as looking at the world through particular lenses, and these lenses have their own set of filters and focus distances, then in principle physics can look at many, many things which normally you would not consider as being within the remit of physics. And a few of these things would be if you look at the physics of sports, for example. So here I just mentioned snooker, curling, and cycling. And in that case, when you are interested in these things, then what the physicist would ask is, how can the rules of physics, as we understand them, give rise to the observed phenomena? Now, what you may notice here is that regardless of whether you're successful or not in this particular endeavor, at the end of the day, usually it won't change the way these sports are practiced, right? So what that means is that looking at the physics of a particular topic is not meant to change the way people have to look at it. However, it's bringing additional information about it to simply enrich our understanding of it. And so here, that's basically what we are trying to do. We are talking about DNA, and we are trying to enrich our understanding of it. So what phenomena related to DNA are we interested in? So it's first efficient compaction. Well, I'm not talking in feet, so I'm talking in meters here. Earlier, Anna Marie talked about 6 feet. I'm talking about 2 meters, about 2 meters of DNA in each human cell. So, or at least almost all human cells. Then, in a sense, it can be interpreted as, or at least looked at as a Rosetta Stone of biology, because it can be read by many different molecular machines, not a single one, but many different ones. And also, it has a context-dependent expression, so it can make some of its information accessible or not, depending on the situation. So then, as physicists, what we can ask ourselves, again, it's quite long, but it's more, how can the rules of physics, as we understand them, then explain the versatile combination of DNA's functional attributes, which are the ones listed there, for example, identified by biologists. So now that this is done, then we can go back to our actual topic, discuss this idea of clothing. And here I will only, I don't have time for 1,000, so I will stick to 3. We'll start by discussing the first outfit. And the first outfit, as you would guess, is actually, no outfit, right? So it's basically a naked DNA. Sometimes people call it bare DNA. In the physics literature, that would be more bare DNA. And so let's have a look at what it is. So here, this particular thing here is this naked DNA thing. This is most of the time what you will actually see. And it's had this particular double helix pattern. Here, in terms of representation, is connected to the remark that Anna Marie made earlier. This would be an experiment-informed representation of DNA. And what that means is that essentially there is some data, like this scattering figure that Anna Marie showed, and somehow we are able to quantitatively link this particular drawing to the structure, to the pattern you saw on the screen in Anna Marie's talk before. So that's what kind of the structure looks like. Now we can try to go at a higher level, and so to do some other searching techniques and talk about the chemistry a little bit of it. Don't worry, I won't talk too much about chemistry, but if you look about this yellow sphere surrounded by 3 red spheres, then these are called phosphate groups. And these phosphate groups tend to be negatively charged in water. And because I'm a physicist, I like talking about electric charges. And so what I will be doing is basically focusing on this aspect. So as I told earlier, I have a particular set of lenses and filters. And so what I will be doing here is focusing on what these charges actually do. So I will simplify the problem then by forgetting about the complexity of what's going on within DNA and focusing on these charges. So I will play with a model where the inside of DNA, with these letters and so on, is just a cylindrical core. And the color here has no particular meaning. By the way, it's just, I thought it was nice, but that's it, for the cylinder. Now, for the double helix, like the blue color means it's negatively charged. Now, before going on to what that means, we need to talk a little bit about what are electric charges, just so that we are on the same page here. So electric charge is basically a property of matter, like size or mass, which we are more familiar with. Now, the electric charge of a material object can be of one of 3 kinds. It's either negative charge, zero charge, or positive charge. Now, the zero charge element essentially does not, let's say, qualitatively does not interact substantially with negative charge or positive charge. So for the time being, we'll put this guy out of the equation and focus on what happens if you've got negative charge and positive charge. Well, as you may know, they tend to attract each other. So the rule here is that opposite charges attract each other. By the way, if some of you have been here last year, some of the 2 or 3 coming slides will repeat some of the things I said then, and that's because I'm talking about electromagnetism. Now, if you want to look at what's going on if they are opposite, if they are of the same charge, then in that, of the same sign, sorry, then in that case they tend to repel each other. Okay, so you would have that charges of the same sign repel each other. Now, one thing we could try to do is to represent this repelling and attracting each other, but in a more, let's say, general way, where I don't need to draw all the possible situations. Okay, so the way to avoid drawing all possible situations is to actually plot what is called a field. So here I'm plotting a field, like a field of crops, essentially, but instead of crops I put arrows. And so this field of arrows would represent the direction of the force that would be felt by a positive particle that would be put around either the blue negative charge on your left or the red positive charge on your right. So indeed, if you put a positive object here, then it will follow the arrows. So in that particular case, it will be attractive and repulsive on this side. Now, this is not enough. Obviously, these arrows are not very elegant, and maybe we can try to interpret this a little bit better. So what we could try to do is introduce, let's say, a more fundamental object to look at. And this object to look at is called the electric potential. And this electric potential consists in saying that these arrows you're plotting earlier correspond to the slope of a landscape, like a mountain landscape, and they are indicating where is downhill, right? So basically these arrows are simply indicating where is the direction to go as downhill as possible. And so you see here that works. So if the landscape is actually very high, then downhill means you go away from the positive charge. And on the other hand, if the potential is very low and then tends to go up as you go away from it, then downhill goes towards the center. Now, to visualize it a bit better, I want to get rid of the arrows, and I will represent things in 3 dimensions. So what I want to do, I start with 2 neutral, so 0 charge, and then I will slowly but surely charge them negatively on your left and positively on your right. So that's what I'm doing here. So as you see, as you do that, you deform the electric potential, and you see basically something increasing there. You've got a hill, and then you've got a valley that is formed by the negative charge. Now that this is clear, we can now talk about DNA. So if I play the same game with DNA, by the way, the rule here is that positive charges follow the arrows, and so they go downhill in the direction of valleys. So let's talk about DNA now. So here this is a model where I imagine DNA with all these phosphate groups I mentioned earlier before, well, it's a bit redundant, where they are initially not charged. And then I will charge them again, as I did earlier, and we'll see how this affects the electric potential landscape. So if I do it, this will give something like this. So you see that there are a few things to be said. First, the height and color scales I'm using are the same as the ones you've seen before. But you see that it's very, very different and much more marked than before. So what we can conclude kind of from this is that bare DNA creates a very steep potential landscape. So one of the things we can say from this is that if electric potential is to molecules what light is to our eyes, then bare DNA acts as a molecular lighthouse, okay, which is basically seen from afar and to which you would run, or molecules would run, or from which they would want to escape, okay? So this is what bare DNA is about, or naked DNA. Now, what would be the related functions? So with colleagues, a few years back now, of course, about 7 years ago, we proposed that it was useful to attract reading and compacting molecules from far away. Okay, that it was an essential features to have. There is also something else, which is that it affects the rigidity of DNA. So DNA is, usually you see it as an helix, but it's actually quite floppy normally. It's moving along, etc. And in principle, you can try to imagine what happens if you try to bend DNA, right? It's what happens if you bend. If you bend DNA, then two of the charges, of the negative charges, will get actually closer to each other. And as a consequence, they would want to repel. So there is basically resistance to bending. And as a consequence, these charges actually affect strongly the rigidity of DNA and make it incredibly rigid for a small molecule like this. And lastly, some people have proposed that it may enable target recognition, so efficient reading of DNA by other molecules. So this is this Rosetta Stone idea I mentioned earlier. All right, so that's for naked DNA. Let's move on to the next outfit. Now, the next outfit is called, I will call it at least, a camouflage gear. Now, why do I call this a camouflage gear? So, we saw earlier this particular simplistic model with all this double helix of charges. Now, as I said quickly before, this double helix is not existing in vacuum like this, right? It's not alone. At the very least, you will have water molecules, okay? In fact, many more than the ones I've drawn here. And you would have so-called counterions. Now, these counterions are just some form of molecules or atoms that have lost or gained an electron. But what matters to us, like the details here are not important for what I'm going to talk about, is that they are red, right? And red means they are positively charged. Okay, that's what I would be interested in here. Now, this system is actually very complicated. Okay, there is all these water molecules around, and then there is these counterions, and there is DNA in principle, which is also wobbly around. So if you want, as a physicist, to model this system, then it becomes first very computationally inefficient. So it takes a long time. You need a huge machine to run for a very long time. And also, it's not clear that you're able to understand what's going on. So to both understand and simplify our understanding, what physicists do is they use a technique called coarse graining. Okay, and this coarse graining approach consists in taking a particular kind of glasses, which basically make us blind to water molecules in that case. So you see they still exist, right? Where the glasses are not, you can see the water molecules. They are still there, but we just put on glasses where we don't see them anymore. Okay, and the only thing we can actually see is the action they have on the ions and how these ions are moving. Okay, so we basically filter out the water molecules from our visible description, and then once we have really put on the glasses and we don't see the water molecules anymore, then we need to extract the new rules. There are new rules of physics that will emerge from it that are followed by the ions in the absence of visible water molecules. Okay, so let's try to see how water affects ions. So first of all, I need to mention that there are many more water molecules than ions. And ions, water molecules, have a tendency to kick around the ions in all directions. And so what happens is that when you decide to put on these glasses, it's as if you were watching a football match, but you didn't see the players, and you could only see the ball, right? And so at the end of the day, what happens is that the motion of the ions appears random. And so the kind of motion you would get would look like this, at least in 2 dimensions, if I limit myself to 2D. So for those people who want to look maybe more into it, it's called Brownian motion. So that's how water molecules affect ion motion. Now, how does DNA affect ion motion? Well, we've seen that DNA is strongly attractive to positive ions, but we also know that counter ions are positively charged. So as a result, we'll have that ions will be attracted towards DNA. Okay, plain and simple, they want to go downhill, so they will go downhill by getting as close as possible to DNA. Now, the thing is that ions also affect DNA. All right, so you see it's complicated, right? So DNA is visible via the electric potential landscape it creates, right? It has this very steep landscape created by DNA, and then that's what makes it very attractive to other positive objects out there. But the thing here is that counter ions also create a particular potential landscape, right? And because they are positives, they would tend to form hills in the electrical potential landscape, while the others don't. So as a result, what we get is that ions will deform the potential created by DNA in this particular way. You see there's a small hump over there, and if you put all these things together, what you get is, listing all this, is that ions move randomly, okay, but they are still being attracted to DNA, and they also perturb DNA's electric potential. So what we get in the end is that ions stay very close to DNA, that's what we are seeing here, but at the same time, DNA's resulting potential is substantially less steep. Now, just in case you don't believe me, you can compare. It's less steep than what we've seen before, okay? So in terms of physics terminology, what we say is that ions actually dress the DNA molecule. And again, to continue in the jargon, the drop in intensity of the apparent potential of this dressed DNA is called electrostatic screening. Now, it is believed by some researchers that up to 90% of DNA's charges can be screened, and then making it essentially invisible to other molecules. And of course, that would be condition dependent. Right, so what would be the related functions? So one function here of this screening mechanism has to do with simply this packaging that Anna Marie mentioned earlier, that I did as well, which has to do with fitting all this DNA to a very small volume. Now, if you've got plenty of charges that the only thing they want is to go as far as possible from each other, and you try to put them all in a single room, then, of course, it will explode. So to prevent that, what you do is you screen DNA's charges. Now, what these curves are showing, if you want to go into details, I'm happy to answer this during questions, but what these curves are showing is that the more screened the DNA is, and the easier it is, well, it's kind of obvious, the easier it is to actually force it into a viral capsid. So here it's related to viruses, but of course it has bearing as well when it comes to packaging into the nucleus of our cells. Now, another thing has to do that we proposed with colleagues 10 years ago, has to do with imagining that this dressing of DNA by counter ions could actually prevent molecules which are reading DNA to read books, if you will, or parts of the genome where it's a waste of time to read. So let's assume you are looking for a book, okay? And instead of simply reading the title of the book, you read all the pages of the book to make sure it's not the book you're looking for. So if you were, so essentially that would be inefficient. At least that's my opinion about it. But so if you were then to switch to a mode where you're relatively far from the book and you simply read the title, then that would be much more efficient to actually find the book you've got to read and actually open it and read it. So here we propose that this ionic cloud dressing was acting as a cushion that would prevent molecules to attach to DNA where basically they shouldn't, or it would be inefficient. Now, it has been proposed as well that this very strong screening could be used and contribute at least to DNA pairing strategy, which is called homology recognition, that plays important roles in repairing and gene shuffling. So, that being said, we can move to the next one. So that was the camouflage gear. Now the next one would be, so the next one would be full protein armor. Okay, so in this particular case, why do I call this full protein armor? So we start with this particular idea. So that's where we left essentially, that there is DNA, and then there is water molecules and contrarians, but that's the very minimum you can get in there. Now, there are other things that can be in a cell, for example, and that would be huge monsters like this one, which are called, here in this case, histone proteins. Now, Anna Marie mentioned proteins before. This is like a specific kind, and I will focus on them for this part. Now, if you have no problem visualizing colors, then you would see that there are 8 different colors here. And these 8 different colors actually represent 8 different subunits, which are the ones called histone proteins. But they tend to come in various forms where they are either paired or in this huge lump that's called the histone core. What I'm interested in, because again, I'm a physicist, is that these guys are actually positively charged. Okay. Now, the fact that they are positively charged will be important. So just so that we can go back a little bit, when we looked at bare DNA or naked DNA, one of the things we said is that the fact that they have these charges all around DNA affect its rigidity and in fact make it very rigid. Now, the thing is that because it makes it rigid, if around DNA you've got contrarians, which are positive, and histone proteins as well, which are both positively charged, then they will actually decrease both DNA's attractiveness, so that's what we've seen with the landscape being much less steep, and its rigidity as well. It will become much more flexible. And as a result, what happens is that DNA ends up wrapping around the histone core to form what is called a nucleosome. So this nucleosome is the beast you see here. So this is a protein core in violet, like purple thing, and then it's wrapped by DNA, and the whole thing, DNA and protein, is called a nucleosome. Okay. Now, what next? Once you've got this nucleosome, what do you do with it? Well, obviously there is not one histone core in a nucleus, so there are many histone cores out there. They will bind with DNA, and they form a string of nucleosomes. And this chain of nucleosome is an assumed, or there is a belief that it will make up a complex hierarchical structure that will efficiently pack DNA. Okay, so this is what would be called the chromatin fiber hypothesis. So here is a short clip that comes from the same source, in fact, as Anna Marie's earlier, about what this thing could actually look like. So this is an artistic animation. So you start with DNA jiggling around, and then at some point it's going to be attacked by histone protein. Now, yeah, you see. So then it's being attacked, and then the whole thing essentially self-assembles into this thing called a nucleosome. Okay, so that's what the nucleosome looks like. I mean, that's what it could look like. Then this nucleosome, you see, you've got a chain of them. They will tend to form, a secondary structure, so-called, which has itself the form of a thread, and that's the so-called 30 nanometer chromatin fiber. And then, well, you probably see where this is going. So then this thing will coil into an additional larger thread, again, a new one, which in turn will also coil into another layer of threads here that you see here. And then, of course, the idea is that that's what makes up chromosomes. All right? So you need to keep in mind this is an artistic representation of what these things could look like. And obviously, all of this is ongoing research to figure out whether these things even exist or not. Right, so what would be the related function of these nucleosomes and chromatin? Well, of course I need to be selective here, but because I called it protein armor, I think it makes sense to talk about protection. So it protects, I mean, at least there is evidence that it protects against DNA damage. And here I mention two sources, so where it could protect from radiation damage, and one where it could protect from iron-mediated damage, which would be more of a chemical nature in that case. Then, in a sense, this chromatin acts as a molecular librarian. So in fact, the level of compaction of this chromatin is not uniform across the DNA in a cell, and it depends on where you are and which cell you're talking about. And this enables basically the chromatin to act as a librarian, which basically grants access or not to some shelves depending on the client. Okay, and so here that's kind of what this chromatin does here, and that's what makes our genome expression so flexible. And finally, there is obviously something that is very exciting in the last decade or so, a bit more. It has to do with this related to trait inheritance. So this is transgenerational epigenetic inheritance. So that's when you can inherit a trait which is not related to the sequence itself, but to how this sequence is expressed via the shape of the chromatin, if you will. Okay, and so all of this is very exciting ongoing research, which enable to go beyond the genetic code paradigm. And so, just to summarize now a little bit. We've seen these three outfits there, and I will try to summarize a little bit in 2 words what these guys are actually saying about what the outfit represents. So naked DNA, well, essentially is very attractive, right? Yeah, I guess you could assume so. Now, camouflage gear, in a sense, could be said as being selectively attractive, right? And then the protein armor, well, it's protectively attractive, right? So it's basically attractive to some part, but basically heavily protecting others. So now we are reaching closing comments. So first, I would say, obviously, it's very tempting to say yet again that we are amazed in front of DNA's ability to use its environment so efficiently, right? But in a sense, that would be a bit unfair, because we could also appreciate the equally remarkable ability of ions and molecules to complement DNA in the role that it has to fulfill. So in fact, if we were to rethink a little bit about the main title of these talks that we've given with Anna Marie, it's Tales of DNA. But in fact, we could call it maybe Tales of DNA and its mates. And then on this, I thank you very much for your attention, and Merry Christmas to all.